Now let's talk about the conduction velocity of the action potential. The two primary factors influencing conduction velocity in nerves are cell diameter and myelination. First, let's talk about the effect of the cell diameter on the conduction velocity. It is very important to note that conduction velocity of the action potential is higher in a cell with greater diameter than in a cell with thinner diameter. If we take a look at these two neurons, the first one has a greater diameter than the second one. Increasing the diameter of a nerve fiber results in decreased internal resistance, so an action potential moves with less resistance. As the consequences, the conduction velocity in a first neuron will be higher than in a second neuron. Talking about the second factor of increased velocity of the action potential, you have to know that there are two types of neurons: myelinated and unmyelinated neuron. It is extremely important to note that the conduction velocity is higher in a myelinated neuron than in an unmyelinated. In order to understand why it is so, let's first talk about conduction velocity in unmyelinated neurons, then I'll explain how myelination of the neuron increases the conduction velocity of the action potential. Let me zoom here a part of this neuron first. So when many voltage-gated sodium channels open and sodium rushes into the cell, causing depolarization of the membrane, this local inward sodium current spreads to the adjacent region and takes the membrane potential of this region up to threshold. Reaching the threshold in the second part of the membrane leads to opening more voltage-gated sodium channels in this next area, and a lot of sodium rushes into the cell and depolarizes this part of the membrane. The sodium which has been rushed into the cell spreads to the third region, opening voltage-gated sodium channels and causes further depolarization and a propagation of the action potential. And of course, don't forget that as sodium rushes into the cell, depolarizing this part of the membrane, voltage-gated potassium channels open and potassium leaves the cell, causing repolarization of the membrane. So depolarization is followed by a repolarization. This is how the action potential propagates and travels to the CNS through the unmyelinated neuron. Now, let's see how the action potential moves through the myelinated neuron and why it is so fast. Let me draw here a part of this myelinated neuron. So these are Schwann cells, which makes the myelination of the neuron, and they are 1 to 2 millimeters in length, and between them we have unmyelinated zones, which are known as nodes of Ranvier. So when a lot of voltage-gated sodium channels open in the beginning of the neuron, sodium rushes into the cell, causing depolarization of the membrane. This local inward sodium current spreads to the adjacent region, usually for 1 or 2 millimeters, which is equal to the length of the Schwann cell. It is very important to note that in a myelinated part of the neuron, sodium conductance is 0 because Schwann cells do not allow sodium to rush into the cell. As I already said, Schwann cells are 1 or 2 millimeters in length, and when sodium reaches the node of Ranvier, where a myelin sheath is not present, it takes the membrane potential of this area up to the threshold. So, reaching the threshold in this part of the membrane leads to opening voltage-gated sodium channels, and a lot of sodium rushes into the cell because here sodium conductance will be high. When a lot of sodium rushes into the cell, it depolarizes this part of the membrane. The sodium, which has been rushed into the cell, further spreads to the adjacent node of Ranvier, causing depolarization of further parts of the neuron. Of course, don't forget that as sodium rushes into the cell, depolarizing this part of the membrane, voltage-gated potassium channels open and potassium leaves the cell, causing repolarization of the membrane. Thus, the action potential appears to bounce from node to node with minimal decrement and greater speed. This type of conduction from node to node is called saltatory conduction. Talking about myelination, it is very important to note that depending on the locality of the myelin sheath, there are two types of cells which can myelinate a neuron: Schwann cells and oligodendrocytes. The Schwann cells are present in the peripheral neurons and can wrap only around one axon. The oligodendrocytes myelinate the neurons in a CNS, and very important point, they can extend to up to 50 axons, wrapping around approximately 1 millimeter of each. The two common diseases, Guillain-Barre syndrome and multiple sclerosis, cause demyelination of the neuron. It is believed that both are autoimmune diseases. The main difference is that in a Guillain-Barre syndrome, the immune system attacks the Schwann cells which myelinate the peripheral nervous system, whereas in multiple sclerosis, the immune system attacks the oligodendrocytes which myelinate neurons in a CNS. Loss of myelin results in disturbance of current leakage across the membrane, and it causes conduction blockage in the neurons.